Good morning. My name is Dr. Abhay Pratap. So today I am going to talk about nitrogen metabolism. Nitrogen is the most abundant mineral element in a plant, the most abundant element in Earth's atmosphere, and the fourth most abundant element in a plant after carbon, hydrogen, and oxygen. Often the limiting nutrient for plant growth. Limiting nutrient means if there is less amount of nitrogen present in the soil, the plants cannot grow normally and they cannot function normally. Nitrogen is in amino acids in form of proteins. They are present in nucleic acids, in genetic materials such as DNA and RNA, chlorophyll, and small molecules. Nitrogen is one of the three major macronutrients found in most fertilizers, as we can see here in this picture. NPK fertilizer, because nitrogen, phosphorus, and potassium are required in large amount compared to other nutrients. Nitrogen is essential for nucleic acid, as we can see in the structure of pyrimidine and purine. In pyrimidine, nitrogen is present at the first and third position, while in purine, nitrogen is present at the 1, 3, 7, and ninth position, as we can see in case of adenine and guanine. Nitrogen can be found in many inorganic forms such as organic nitrogen, urea, ammonia, ammonium ion, nitrogen, nitrous oxide, nitric oxide, nitrite ion, nitrogen dioxide, nitric acid, and nitrate ion. The nitrogen compounds are present in various oxidation states ranging from -3 to +5, means highly reduced to highly oxidized state. The oxidation of ammonia to nitrate is known as nitrification. This is an aerobic respiration, means oxygen is required in this process, whereas reduction of nitrate to ammonia is called as denitrification. And in this process, there is a reduction of nitrate to ammonia, so there is no requirement of oxygen, and this is completely anaerobic reaction. Plants are an important part of the global nitrogen cycle because only plants can assimilate the nitrogen that is obtained from the atmosphere. In the atmosphere, nitrogen is present in the form of gas, so which cannot be taken up directly by the plants. So what happens when the plants dies or when the animals eat the plants, the nitrogen is transferred from the plants to animals, and once these animals dies, then they are decomposed and the ammonium is oxidized to nitrate by the help of nitrifying bacteria. And again this nitrate is taken up by the plants and assimilated in the plants. Now there are very interesting questions. How do plants optimize their uptake and utilization of nitrogen? How is nitrogen taken up into the plants? How is inorganic nitrogen assimilated into organic molecules, such as we eat the seeds, fruits? So how they are converted from the inorganic to organic molecules? We'll understand it. How do plants sense local soil nitrogen levels and plant nitrogen status? How do plants respond to nitrogen deficit? How do they maximize uptake through their roots? How do plants remobilize nitrogen to optimize nitrogen utilization? These are very interesting questions. Let's understand them. The nitrogen metabolism, its uptake, assimilation, and remobilization. So nitrogen is taken up by the plants in form of nitrate, and this nitrate is reduced to ammonium. So it is reduced by nitrate reductase. This is two-step reaction. In first step, nitrate is converted to nitrite, and then nitrite reductase convert it into ammonium ion. Or other way, ammonium can be directly taken up by the plants. So once ammonium is taken up by the plants, it is converted to glutamine with the help of glutamine synthetase enzyme. And again, this is known as amino acid, which can be incorporated into different nitrogen containing compounds. Again, from this glutamine, with the help of enzyme glutamine to oxoglutarate aminotransferase, it is converted to glutamate, which is another important amino acid. And this is how its cycle goes on with the help of GS-GOGAT enzyme. And this ammonium is also remobilized through the amino acid recycling and photorespiration process. Most plants take up most of their nitrogen as nitrate. Many prokaryotes oxidize ammonium, so soil ammonium levels are often low because this is oftenly oxidized by the nitrifying prokaryotes. So as we can see in this reaction, ammonium is sequentially oxidized to nitrate and then again oxidized to nitrate with the help of these nitrifying prokaryotes. And in this process, energy is released. The plants use energy to reduce ammonium for assimilation into organic compounds. So in this case, the nitrate is taken up by the plant, is reduced to nitrite, and to again in next step it is reduced to ammonium. So in this process, energy is required in form of ATP to convert it into organic compounds. Again, very important point. Plant preferences for ammonium versus nitrate vary by the species, other metabolic processes such as temperature, water, soil, pH, etc. Plants have specific transporters for nitrate, ammonium, and other nitrogen forms. Means there is no single transporter which can allow the take-up of nitrogen compounds. These are very specific transporters. For ammonium, there is different transporter. For nitrate, different transporter. And for urea, there is different transporters. And these transporters are also two types. One is high affinity transporters, we call as HATs, and low affinity transporters known as LATs. So what is the difference between high affinity transporter and low affinity transporters? So how they work actually depends on the availability of nitrogen in the soil. If there are more nitrogen, then low affinity transporters allow to uptake of nitrogen compounds. But when there is very less concentration of nitrogenous compounds present, then high affinity transporters are responsible because their affinity is very high. They can allow a very small concentration of uh, nitrogenous compounds, whether it is ammonium or nitrate or urea. A major nitrate importer was the first cloned in as CHL1, NRT 1.1 and PF 6.3. The first nitrate transporter was identified using a genetic selection for chlorate resistance. What is chlorate? Chlorate is actually an analog of nitrate. It is having the similar structure as of nitrate. But if nitrate is reduced to nitrite, it is not toxic for the plant. But when chlorate is reduced to chloride, it is toxic to plant and plant cannot survive in presence of chloride. So how scientists prove this? They have made a mutant of chlorate uptake, means that plant cannot take, take up the chlorate. So once they made this plant, then they made a simple assay. They have grown these plants on the chlorate medium. Chlorate medium means they will add the chlorate in MS agar. That is the uh, medium where plants can grow. So when wild type plants were grown on chlorate medium, it was dyed. Why it was dyed? Because chlorate is taken up by the plants and that is reduced by the nitrate reductase. And then it was converted from chlorate to chloride. And chloride is toxic. So this plant was dyed. In next case, this plant was nitrate reductase mutant. Means nitrate reductase enzyme is not working here. So what will happen? Chlorate will be taken up by the plant, but it is not reduced to chloride. So it will not affect the plant. Plant is happily surviving. In third case, where the chlorate uptake mutant is used, here what will happen? Chlorate will not enter into the plant. Why? Because the chlorate uptake, it is a chlorate uptake mutant. There is chlorate transporter is inactive. So what will happen? Chlorate will not enter into this plant and this plant is also surviving well. Thank you for listening.